All right, so welcome to week 11, BIOH122 webinar. So this is going to be session 19. This is our last session on the renal system. So we will be talking about the last few mechanisms and concepts involved in formation of urine. And we will also look at the fluid balances in our body and the structure of bladder and ureters and urethra. So the rest of the renal system. Okay, so we will start now. So I'm just going to bring up my session 19 PowerPoints. Right. So last session on the urinary system, right? So what is going to be the focus for today's session? Primarily, we are going to look at the function of the loop of Henle, which is the part of the nephron, right? So we will be looking at how it contributes to forming the concentrated or diluted urine. So that's going to be the first point. So production of concentrated and diluted urine. Then I'll briefly talk about the renal functions and how we can use different tests to look at the renal function. The next part of the session will be looking at the structures of ureter, bladder, and urethra. And while we are looking at these structures, we will also talk about the maturation reflex, that is the reflex that helps us to void the urine out of the body. And then that will be the first part of our session today. And the second part is all about fluid balance in the body. So we're going to look at the different buffer systems and the regulatory mechanisms, the role of different hormones in maintaining the fluid in our body, basically. Okay. A lot of these concepts that you will discuss today will be useful to answer the questions in your lab 3. Which is, I think it's next Wednesday. So this Wednesday, sorry, coming Wednesday. So the scenario and everything, when you have a read through it, you will realize that a lot of questions that are in the scenario, they're based on all this osmolarity and concentration of urine and concentration of electrolytes in the body and things like that. All right. So before I start with the session, does anybody have any questions from the last session that we did on Wednesday? Any questions around tubular reabsorption and tubular secretion? All right. Okay. So we can then start. All right, so you remember in the last session, we talked about tubular reabsorption and tubular secretion. So just kind of a recap for that, I just want to bring up a diagram to kind of recap that because that is linked to what we're going to do today, okay? I did ask you to make your own diagram with the nephron to kind of summarize what are the things reabsorbed and what are the things secreted in the nephron, right? So when we talk about reabsorption, it is about substances moving from the nephron into the peritubular capillaries. And when we talk about tubular secretion, it's about substances moving from the peritubular capillaries back into the nephron, into the tubes of the nephron. So proximal convoluted tubule, loop of Henle, distal convoluted tubule, or collecting duct, right? So if you look at this diagram here, you can clearly see, basically see that majority of reabsorption of substances is taking place in the first part of the nephron, which is the proximal convoluted tubule. This is the part that reabsorbs about 65% of water, almost 50 to 55% of your electrolytes or the salts that we call them. So like sodium, potassium, chloride, bicarbonates, all of this, right? So you see that sodium, about 65% of sodium is reabsorbed, potassium, same, chloride, about 50%, 90%, 80 to 90% of bicarbonates are reabsorbed. So all of the electrolytes and salts are pretty much reabsorbed in the proximal convoluted tubule. Okay, glucose, 100% of glucose, 100% of amino acid is also reabsorbed in the proximal convoluted tubule. Why is it important to note this? Is because this reabsorption, the amount of things that are taken back into the peritubular capillaries, will decide what will be the gradient, osmolarity gradient, that is a difference between the concentration of solutes inside the filtrate and difference between the concentration of solute in the peritubular capillaries. So that gradient will decide the flow of water and the formation of urine, whether it will be concentrated or diluted, right? So proximal convoluted tubule will set up the gradient for osmolarity. Okay, there are few things that are secreted in this part of the nephron, like creatinine is secreted here, some of the hydrogen ions are secreted, ammonia is secreted, and urea is secreted back into the renal tubule. So tubular secretion, four things are secreted in the proximal convoluted tubule. So this is the proximal convoluted tubule where my cursor is at the moment. And so majority of the reabsorption is taking place in this part, and few things are put back into the renal tubule, which is basically hydrogen ions, ammonia, urea, and creatinine. Okay, as the filtrate descends into the loop of Henle, again the reabsorption continues. Now another important thing to notice about the loop of Henle is that the first part of the loop of Henle, which is the descending limb of the loop of Henle, it is permeable to water, so it will reabsorb water and put it back into the peritubular capillaries. But the last part of the loop of the Henle, or the remaining part, that is the ascending limb and the remaining part from the DCT and collecting duct, these are pretty impermeable to the loop of Henle. So absorption of water, reabsorption of water into the peritubular capillaries will decrease as the water goes from the descending limb to the ascending limb and then into the distal convoluted tubule and the collecting ducts. Okay, by the time it reaches the DCT, distal convoluted tubule, and the collecting duct, these structures are practically impermeable to water, so no more water reabsorption takes place here. Okay, this is the basic concepts. Now we use these concepts to understand how concentrated and diluted urine are formed. So let us go back. Okay, let's start with the session. So all this concentration of urine, the formation of urine, is actually regulated by some of the hormones, particularly to look at the antidiuretic hormone. Anti means opposing. Diuresis is urination. So this hormone is something that is opposing urination. So whenever we want to form a concentrated urine, we need to reabsorb most of the water from the renal tubules. Usually, as I explained earlier, the ascending limb of the loop of the Henle, the DCT, and the collecting ducts, they do not absorb as much water back into the blood capillaries, right? But if your body needs that extra water, then it will release antidiuretic hormone. So the signal for release of antidiuretic hormone from posterior pituitary is basically the salt concentration in the renal tubules. When the in the plasma, so blood plasma. So when the concentration of the salts in the plasma increases, when the concentration of sodium or potassium in the blood plasma increases, this sends a signal to the osmoreceptors in our hypothalamus. Hypothalamus is the organ that is producing antidiuretic hormone. So the neurosecretory cells in your hypothalamus will release antidiuretic hormone into the posterior pituitary when the osmolarity of the plasma is high. So when the salt concentration in the blood plasma is high, posterior pituitary is stimulated to release antidiuretic hormone, and this antidiuretic hormone, it works on the collecting duct. It works on the principal cells of the collecting duct. And how it helps to reabsorb water? It puts in extra channels. It puts in aquaporins, aquaporin 2, which are special channels that are only inserted into the principal cells when ADH is released in the blood. So when there is ADH in the blood, Then ADH stimulates insertion of aquaporin 2 in the principal cells of your collecting duct, and they allow water to be reabsorbed through diffusion, right? So water will always follow a passive process. It will always follow from a higher concentration to the lower concentration. In the in the distal convoluted tubule and collecting duct, the concentration of water is high. Right? Remember, because they are impermeable to water. So when aquaporin channels are inserted into these tubular cells, water will go from the higher concentration to the lower concentration. So water will go from the renal tubules into the peritubular capillaries. So what will happen to the urine? The urine that is being formed in the collecting duct, that is being accumulated in the collecting duct, it is actually losing water. So it becomes more concentrated. So that type of urine will be very concentrated urine. Okay. So that is basically how the concentrated urine is formed because of the antidiuretic hormone. So we will talk about this in a little bit more detail. But just giving you, this is a recap of the previous session, like how hormones regulate the formation of urine, right? So remember that main hormone that regulates the formation of urine is antidiuretic hormone, and to some extent, yes, aldosterone also will help in some cases, particularly when the blood volume is low and there is severe dehydration and blood pressure is low, then yes, aldosterone also comes into play. Okay, so when we talk about the diluted or concentrated urine, see, basically what is a diluted urine? A diluted urine means its osmolarity, that is the composition of salt or solutes in this urine, is very less compared to that in the blood plasma. If we compare the blood plasma, the amount of salts present in the plasma is basically about 300 milliosmoles per liter. So that is the concentration of all the different types of salts. We're not only talking about sodium or potassium, but we're talking about all the different types of electrolytes together, molecules together in the blood plasma. They amount to approximately 300, strength of approximately 300 milliosmoles per liter. So that is the osmolarity of the plasma. Compared to that, osmolarity of a diluted urine is almost 1/4 of that. So very, very less. So only about, you say, about 60 to 70 osmoles per liter, 65 to 70 milliosmoles per liter. So very, very less. So what it means is that in a diluted urine, you have very few solutes compared to the concentration of water. So you have a lot of water with very less solute. How this happens and why this happens. So that is basically depending upon the nephron that is participating. Okay. Now, you know, when we looked at the nephrons and the structure of the nephrons, I think in the first session we talked about this. I did mention that there are two types of nephrons. Okay. You have cortical nephrons and you have medullary nephrons, right? So cortical nephrons are the ones where the loop of Henle is pretty much in the first part of the medulla. It doesn't go much deeper into the medulla, and almost most of the nephron is in the renal cortex area. Okay, renal medulla is always going to be very, very salty. As you go down deeper into the medulla, renal medulla, the concentration of the salts in the interstitial fluid, so in the fluid in outside the nephrons, is always going to be very, very salty. It's going to be much more high osmolarity. Okay, so here in this diagram that I've put up here, you can actually see the changes in the osmolarity. This one is showing you a cortical nephrons. How do I know that it's a cortical nephron? I look at the loop of the Henle. The loop of the Henle, so this part of the cortical nephron is usually very short. It's not long, it's very short, and it ends in the first part of the medulla. So top portion of the medulla, doesn't go much deeper into the medulla. So if you look at the osmolarity differences that is happening, see what is happening. So here let's start with the efferent arteriole and the filtrate being formed. Okay, so as the blood is coming into the efferent arteriole, into the glomerulus, you can see the blood coming in, right? As it passes through these glomerular capillaries, lot of water and solutes are filtered in this cup, which is the glomerulus, right? So this filtrate that is formed here, the filtrate will have the osmolarity with about 300 milliosmoles per liter, which is exactly same as that of the plasma in the blood that is flowing into the glomerular capsule, right? So at the beginning, the osmolarity is the same, right? As the things pass through the proximal convoluted tubule, 65 to 70% of water is also reabsorbed along with the same amount of salts, right? So in the proximal convoluted tubule, what we are seeing is obligatory reabsorption of water. Now, when we were talking about tubular reabsorption of water, I mentioned that water follows two methods of reabsorption, right? There is an obligatory reabsorption that is happening, which is basically where the water molecules follow the solutes. So wherever the solutes are going, the water molecules are going in that direction. So that's an obligatory absorption, okay? And there is a facultative absorption of water. Facultative reabsorption means water is specially reabsorbed, irrespective of what is happening to the solutes. We are specifically reabsorbing water molecules, and that is what you see when antidiuretic hormone is released, right? So antidiuretic hormone forces facultative reabsorption of water, while in absence of antidiuretic hormone, water is just obligatory following the solutes, right? Water follows glucose, water follows sodium, water follows proteins, right? So what is happening here in the proximal convoluted tubule, the osmolarity of the tubule in the first part of this PCT is not changing. It was 300 when the filtrate was formed, and throughout the PCT, it is still remaining at 300, right? This is not changing. Why it is not changing is because the amount of solutes that are being reabsorbed and the amount of water that is being reabsorbed, it is balanced out. Same quantity of water is being reabsorbed as the solutes. So the concentration of the filtrate is not changing. Does that make sense?
Yes.
Yeah. So that's basically in the PCT. But then as it starts going into the descending limb, as the filtrate passes into the descending limb, things change. Because as it goes into the descending limb of the loop of the Henle, absorption of the solutes decreases, right? So the concentration of solutes in the renal tubules will increase. Water is still being reabsorbed in the first part of the loop, right? So water is leaving, solutes are still remaining in the tubules. So you can see that the concentration of the salts is increasing. So osmolarity is increasing as you go down further into the descending limb. Okay, right at the end of the loop of Henle, in a cortical nephron, the osmolarity will be about 900 milliosmoles per liter. If you look for the same process in a medullary nephron, where the loop of Henle is much longer, the osmolarity will be much, much higher, almost 3 times higher than what you see in the cortical nephron. Okay, so when body is forming diluted urine, it is using cortical nephrons. When it needs to form concentrated urine, so when it needs to conserve water, it will use the medullary nephrons. Okay, so as the filtrate passes from the PCT down into the tubule, you can see that the osmolarity is gradually increasing, right? So from 300 to 350 to 550 to 750 to approximately 900 milliosmoles. This is happening because in the descending limb, the absorption of solutes is comparatively less than absorption of water. Absorption of water is still happening. So about another 15% of water is still being reabsorbed in this part of the loop of Henle. right? Compared to that, the amount of salts that are being reabsorbed is very, very less. Only 5 to 10% of the salts are being reabsorbed. And lot of things are also being put back into the loop of Henle, into the descending loop. Okay, so let's go back to that particular diagram to kind of bring this into the context. I'm just going to scroll down to bring that diagram up. So if you see the loop of Henle, right? So in the loop of Henle, urea is secreted, sodium chloride, potassium is reabsorbed, water is mainly reabsorbed. So a lot of things are being reabsorbed in this loop, right? Particularly in the first part of the loop. First part of the loop is mainly for the water reabsorption. The second part is, yes, for the sodium and chloride and all the other things. So we'll talk about that now. So what happens then? As the filtrate, let's go back to our diluted urine, right? So when it is going down into the descending limb, what is being reabsorbed? Water is primarily being reabsorbed, right? Water is being taken out of the renal tubules and it's put into the peritubular capillaries. As the filtrate passes through the ascending limb of the loop of Henle, ascending limb of the loop of Henle is impermeable to water. The absorption of water is very restricted in this part of the loop. So what happens? Water remains in the tubule, but sodium, potassium, chloride, bicarbonates, these are the things that are leaving the renal tubules. These are actively pumped out. There is active reabsorption of the solutes. So what happens to the osmolarity? See how it is dropping. From 900, it drops to 550 to 350 and almost 150. By the time it reaches the distal convoluted tubule, osmolarity has dropped to 100 milliosmoles per liter. So very, very less compared to the plasma, right? And why did this happen? This happened because this brown structure that you are looking at, the thick brown structure, the thick wall structure, this is impermeable to water molecules under normal circumstances. So water remains in the renal tubules. It is not reabsorbed. So it dilutes the urine. Does that make sense? Yeah, okay. So this process will continue even in the collecting duct. Same thing will happen. Even in the collecting duct, the walls of the collecting duct are impermeable to water. So water will remain in the collecting duct, and that water will then become the part of urine and will be excreted out of the body. So such a urine will have much more water compared to the solutes. Okay. So here you can see this is the collecting duct, and these are the DCTs, distal convoluted tubules from different nephrons draining into the same collecting duct, and they are all doing the same process. As far as cortical nephrons are concerned, cortical nephrons are important for diluted urine formation. So most of the time these are the ones that are working, and they are making the urine much more diluted because they are conserving water within the renal tubules. Okay, so that is the first part. Clear, everybody? Yes, that's one. Okay, so now we move on to the next one, which is basically looking at what happens in the medullary nephron. So what happens when you want to form concentrated urine? Okay, so when you want to form concentrated urine, so concentrated urine again is a urine where the osmolarity of this urine is very, very high, 4 times that of the plasma. So if your plasma osmolarity is say 300, then the osmolarity of the concentrated urine will be 4 times 300, which is 1200 milliosmoles per liter. So very, very high. So a lot of salt is there in the urine with very less water. So when will you have to do that? When will the body do that, or when will the body form concentrated urine? Well, the body forms concentrated urine when your blood volume or your plasma volume is low. So in conditions where there is severe loss of blood, or where there is dehydration, or when the body needs more water in the cells rather than out of the body, in those situations, the concentration of the urine will increase. Body will try to reabsorb as much water as it can. Okay, and to do this, it will use hormones like antidiuretic hormone. So what happens here? Again, the principle remains the same, that the water will follow its osmotic gradient. It will go from a higher concentration to the lower concentration. That doesn't change. But what changes is that with the help of antidiuretic hormone, you are inserting more channels, more aquaporin 2 channels, water channels into the distal convoluted tubule and collecting duct, primarily into the collecting duct, because that's where the principal cells are located, right? Okay. So in order to form the concentrated urine, so here I'm going to just skip a few slides to bring up the particular diagram, right? Now here you are looking at the juxtamedullary nephron. So you're looking at the longer nephron. See this one, if you compare this one to the previous one that you looked at, look at the loop of the Henle. Look at the length of the loop of Henle. Loop of Henle here is much more longer, right? It is going much deeper into the medulla. Right? So as I said, as you go down deeper into the medulla, the concentration of salts in this region increases. So the interstitial fluid becomes more and more saltier as you go deep down into the medulla. And this is because of the active pumping out of the salts by the ascending limb of the loop of Henle. So let's have a look at the osmolarity gradient here, so to understand how the concentrated urine is being formed. Okay, so if you look here, we start with the PCT, right? Nothing changes in the PCT. PCT, you had the osmolarity of 300 in the filtrate, and it remains 300 till the things are in the proximal convoluted tubule, right? But as the things go down into the descending limb, you see that by the time it reaches the end of the loop of Henle, the osmolarity has increased to approximately 1200 milliosmoles per liter. Reason for this is the length of the tube. The longer the tube, more chance for the water to be reabsorbed. So you can see here water is leaving the renal tubules, right? So as the water is leaving the renal tubules, the concentration of solutes in the renal tubules is increasing. Longer the distance to travel, more chances for the water to be reabsorbed, because we know that the descending limb of the loop of Henle is permeable to water, right? It is absorbing water. So it will do that passive absorption all the way down. Okay. Now, when the loop of Henle is so concentrated, when the solute here is causing such a high concentration of osmolarity, as it passes up the limb of the loop of Henle, things will change drastically. So from here, from 12,000, you can see as it is going up, the osmolarity is reducing gas to 800, 600, 400 and down further, right? Now, at the same time, sodium and potassium are being actively pumped out. So it is still continuing to make the solutions very, very salty. Right? If there is no antidiuretic hormone, this will continue and the urine will be formed, which will be slightly concentrated, right? It will be concentrated because the osmolarity was very high. So it won't be as diluted, it will still be concentrated. But when ADH comes into action, things change a bit more. With antidiuretic hormone coming into the secretion, antidiuretic hormone opens up those aquaporin 2 water channels. So what is happening? So osmolarity was reducing, but now the osmolarity is increasing. See, distal convoluted tubule, the osmolarity went from 100 in the ascending limb to 200 in the DCT to 300 in the collecting duct. By the time it reaches the papillary duct, so the lower part of the collecting duct, the osmolarity has become 1200, almost same as that you had in the loop of Henle, right? So you can see that the urine is becoming more and more concentrated, and this is because as the thing is passing through the collecting duct, water is leaving the collecting duct. Solutes are still remaining in the duct, but water is leaving out. And at the same time, urea is also being added into the tubules. Tubular secretion of urea happens in the renal medulla. This is called recycling of the urea. Generally, kidneys will allow urea to pass out, right? About 54% of urea needs to be eliminated from the body, and that's what they will maintain. But when you want to conserve water, what you want to do is you want to give out as much urea from the body as possible. So in a concentrated urine, that strong smell of that urine comes from that excess of urea which is being put back into the renal tubules, and this is just to maintain the osmolarity of the tubules so that the urine concentration can be created, like concentrated urine can be created and water reabsorption can be maintained. Right? This mechanism of increasing the osmolarity as you go down further into the medulla and maintaining the composition of urine is all based on the direction in which these fluids are flowing. So that mechanism is called counter current mechanism. So we will have a look at the counter current mechanism in a minute. Okay. See the change in the osmotic gradient from 300 down to 1200. Water is still following the osmotic gradient. So water is still doing passive transport. It is going from a higher concentration to a lower concentration area. That is what it is doing, right? But what is facilitating that passive transport? The active transport of salts and the channels, aquaporine channels that are inserted into the collecting duct, and secretion of urea back into the renal tubules. These are the things that are facilitating the direction in which water is moving. Clear?
So will you only use these nephrons when you dehydrated them?
Yes, yes. When you are dehydrated or when your overall water consumption throughout the day has been very less, in that case, yes.
Otherwise, in normal situations, you will use the other nephrons that have the shorter loop of Henle. Is that correct?
Yes. Okay. Yes, that's correct. Yes. So most of the time you will just be using the cortical nephrons, so the shorter ones, and only when your overall fluid intake is less or you are severely dehydrated, that's when you will be using the medullary nephrons.
So the interstitial tissue will be even lower, like if it's 1,200 inside the duct, outside will be even lower for the water to move out. Like in the duct,
for that one.
So in the duct, you have the 1,200
yes,
water pressure or water balance. So in the outside, in the interstitial tissue, will that be even lower for the water to still move out?
Yes, yes. The concentration of water in the interstitial tissue will be less, because you see here in the ascending limb, the salt, the green arrows, they are showing you the active pumping out of the salt. So what the renal tubules do, they actively pump out the salt. So they increase the concentration of salts in the interstitial fluid. So therefore the fluid is very salty. So the water from the tubules will go into that salty solution. Yes. Because the concentration of water outside is less.
Okay, thank you.
Yeah. Okay. All right. So we can then look at the counter current mechanism, right? So the things that drive this flow and how the osmolarity increases is based upon two mechanisms. First is the counter current multiplication of the fluid that is flowing through the renal tubules. So it depends upon the direction in which things are flowing. Counter current means one thing is going down, other thing is going up, right? So we are talking about the flow of filtrate in the loop of Henle. If you look at the loop of Henle, I'm going to bring this direction up. Right? So if you look at the loop of Henle, in the descending limb, this is the direction from up, down, right? So it's going in this direction. The things are moving in this direction. In the ascending limb, the things are moving in the opposite direction. So when the things are moving in this direction, the osmolarity of the solutes is increasing. When the things are moving in the upward direction, the osmolarity is decreasing because water is being retained in the normal circumstances, right? So longer the distance for the things to travel, more osmolarity gradient will be created because there will be more reabsorption of water happening along the length of the tube. While this is happening, the peritubular capillaries around the tubules and the vasa recta, which is in between the loop of Henle, right? So they are also maintaining the exchange of solutes. So that is the counter current exchange mechanism. So amount of solutes being exchanged between the blood capillaries and the renal tubules. So as you can see here, water is leaving the renal tubules into the interstitial fluid. At the same time, vasa recta is picking up sodium and chloride here in order to maintain the osmolarity in the vasa recta. Because again, for water to go into the peritubular capillaries, you need the concentration of water here to be less compared to that in the interstitial fluid, right? Only then it will go from the interstitial fluid into the blood capillaries, right? From high to low. So in order to maintain that, even the vasa recta is giving out the water and taking up sodium chloride. So it is inside these vasa recta, inside these capillaries, it is creating an environment where the water is very low. And so water quickly follows that passive transport. So here you can see that in the ascending part of the vasa recta, water is quickly going in while the sodium and chlorides are going out. Okay, so these two mechanisms, the counter current multiplication factor and the counter current exchange of the salts, they will drive the osmotic pressure, they will drive the osmolarity in the nephrons, and they will help to create either a diluted urine or a concentrated urine. Clear?
Feels like a lot of loss of energy to pump all those sodium into the blood and to just pump it out again.
Yeah, well, sodium here in the vasa recta, in the descending limb, sodium is just flowing from a higher concentration to lower concentration. So it is not using energy there. But yes, in the ascending part, it is using energy. Same here, in the ascending part, it is using energy, not in the descending part. Descending part, it will just follow the passive transport, facilitated diffusion using ion channels, right? But yes, in the going upwards, yes, it uses the active energy. Clear?
Yes, thank you.
All right. So that helps us to concentrate the urine. Now, why is this important? So this is where the clinical application comes in. And also in your lab also, a couple of scenarios are around this concept of what happens when you take diuretics, or what happens when you take alcohol, or what happens when you have too much salt in your diet. Okay, so if you see clinically, these things, understanding these mechanisms are important because drugs like diuretics, they alter these mechanisms. Diuretics, they increase urine formation. So what they do, they reduce the renal reabsorption of water, particularly in the descending limb of the loop of Henle, because that is where whatever reabsorption of the remaining water is happening is taking place. The ascending limb is already impermeable, so no reabsorption or very less reabsorption is happening in that section anyways. Okay, so it is working in the loop of the Henle, so descending limb of the loop of the Henle. Some of the diuretics, they work on the DCT, yes, so in order to form more urine in the body and allow you to eliminate ions and water from the body. Okay, alcohol is a natural diuretic. It increases secretion of it inhibits secretion of antidiuretic hormone. So it affects the hypothalamus and reduces the secretion of antidiuretic hormone. So no antidiuretic hormone, therefore more diluted urine is formed. Okay. So that is basically where the clinical application comes in. This mechanism will also be useful to explain why, if you have very salty diet, right, what will happen to the water exchange? Because when you have too much salt in your plasma, the osmolarity of the plasma will be very, very high. So normal osmolarity is 300. You have extra salt in your body, then the osmolarity will be much, much higher. And if that is much, much higher, then water will not easily leave because it wants to balance out that osmolarity. Yeah. Okay, so we talk about the different tests that are done for the evaluation of the kidney function, right? The most important and most frequently used test is urine analysis, urine sample analysis, right? So when we talk about the urine analysis, there are two types of analysis that are done. There is a macroscopic analysis, so where you look at the color of the urine, the physical properties of the urine, the volume of the urine, and the chemical composition of the urine. And we can also do a microscopic analysis where we actually look at the microscopic structures, features of the urine that is collected. So we look at the presence of any cells, presence of any micromolecules, presence of casts, for example, things like that. So red blood cells, white blood cells, casts or parasitic fragments and things like that in the urine. So that's what we measure in microscopic examination of the urine. Okay. Apart from that, in order to evaluate how well the kidneys are performing, we can also do the blood test. We can do the serum analysis or the plasma levels of certain substances that are thought to be eliminated from the body by the kidneys. Almost majority of it is eliminated. So particularly creatinine, it's very, very important. Now, when we talked about creatinine, I mentioned that 100% of creatinine that is circulating in your plasma will be eliminated by your kidneys, right? So the plasma creatinine level generally remains very, very low. It almost remains under 1 moles per liter. So 1 milli moles per liter. So very, very less. Okay. But if the creatine levels are rising in the blood, then it means that kidneys are not eliminating all of the creatinine that is ending up in the filtrate. So that can be a suggestion that the kidneys are not performing their function optimally. So that is called the renal plasma clearance. So what we are basically looking at, we are looking at how much creatinine was present in the blood and how much of that has been cleared in the urine. So we look at the plasma concentration of the creatinine, we look at the creatinine in the urine, and we use those numbers to calculate the clearance, plasma clearance of the creatinine. It's called creatine clearance test, CCT. Okay. So just normal properties of urine. Okay. Normal properties of urine under normal circumstances, when the person is drinking, say, about 2 liters of water in a day, whatever fluid is, or even including the fluid diet and everything, whatever fluid is taken into your body, almost 99% of that is reabsorbed, and about 1% is eliminated out of the body, right? So that will, it is actually balanced. So whatever fluid you take, it is the amount of fluid that is eliminated is pretty much balanced, right? So volume of the water depends upon the volume, sorry, volume of the urine depends upon the volume of water or fluid that has been taken throughout the day. So assuming that a person drinks about 1 to 2 liters of water throughout the day, volume of urine in 24 hours is about 1 to 2 liters. Of course, this will depend on the person's water intake as well. Okay. Color of a normal urine is light yellow, not very concentrated, not very diluted. So light yellow or amber color. If there is a concentrated urine, it will be darker in color, so it will be golden yellow or dark yellow color. If it is very diluted, it will be clear, colorless. Okay. Turbidity. Water generally is urine generally is transparent, so because it doesn't have any protein in it, right? 100% of protein are reabsorbed into the blood, so it doesn't lose any protein in urine. So therefore, urine generally is transparent. If there is turbidity in urine, if it is cloudy or if it turns cloudy after standing in the container for a while, then it means that there are proteins present in the urine. Okay, so presence of any proteins will make it turbid or cloudy. Odor, generally urine has a very mild aromatic odor like in ammonia. This odor will increase if the concentration of urea in urine is higher. So in concentrated urine, it will be very strong odor. Okay. Urine is always slightly acidic, so pH of the urine remains very, very low compared to that of the plasma. Plasma is slightly alkaline, right? So pH always remains slightly over 7, so 7.2 to 7.3. Urine, the plasma urine pH will remain around between 4.6 to 6 average. Okay, if it becomes alkaline, then yes, it goes up to 8. Okay, specific gravity, which is the amount of solutes in the urine, right? So specific gravity of urine is pretty much similar to that of normal water, so it remains around 1, at the most 1.035. Okay. If the concentration of solutes increases, so when we're looking at the concentrated urine, the specific gravity will be significantly high, at least 2 to 3 times higher. So these are the normal properties of the urine, normal physical properties, you can say, in the urine. In normal urine, you will never find glucose, you will never find ketones, you will not find proteins. You may find few epithelial cells, you may find occasionally few WBCs, but you will not find red blood cells, you will not find casts. These things are not present in a normal urine. Okay, so when things like glucose, protein, or ketone bodies are present in the urine, that is definitely something abnormal happening with the kidneys or the urinary system. These are the terminologies used for presence of abnormal substances in the urine. Now, some of these can be a part of your multiple choice questions in your final exam, but mainly it's just about understanding what each terminology means, right? So anything, when we talk about anything being present in the urine, we use the word urea. Like for blood, we use the word amia. So anything present in the blood is called amia, like glucosamia, proteinemia, like that, bilirubinemia. So when we talk about urine, it is all about urea. So glucosuria is presence of glucose in the urine. Proteinuria is presence of protein in the urine. Hematuria is presence of heme, that is blood in urine. Bilirubinemia, or sorry, bilirubinuria is presence of bilirubin in urine. Okay. Hemoglobinuria is presence of hemoglobin in the urine. Pyuria is presence of pus or white blood cells in urine. Okay, so what can cause all these things? These are just some conditions given. A few examples are given. You don't have to worry about them at the moment. Don't worry too much about this, just for your knowledge. So like for example, glucose increases in diabetes mellitus. Proteins will increase when something is wrong with the kidneys, so like glomerulonephritis or nephrotic syndromes or high blood pressure and renal diseases and anything like that. Ketones will generally accumulate when there is too much sugar in the blood. So diabetes, sugar breakdown forms ketone bodies, so they will accumulate in the blood and therefore end up in the urine. Hemoglobinuria occurs in severe blood transfusion reactions or anemias and burns and things like that. Bilirubinemia, so generally bilirubin does not end up in urine. It is reabsorbed by your liver, and only when there is liver diseases or liver infections like hepatitis, for example, then yes, excess of bilirubin will end up in the urine, and that's why the color of the urine will become much more darker. Okay. Hematuria, which is blood in urine, that can be a sign of simple things like urinary tract infection or renal stones or things like that, or even more severe things like say cancer, renal cancer or anything like that. Okay. Pyuria generally means infection. Any pus collection anywhere in the body is infection. So it's just for your knowledge. You don't have to worry too much about remembering these possibilities, but just kind of giving you an outline. Right? Okay. Then we move on to understanding the maturation reflex and the structure of ureter, bladder, and urethra. Okay, so once the urine is collected in the collecting duct, it passes from the collecting duct into your uh, papillary duct, and from the papillary duct into your minor calyces and major calyces, then through the renal pelvis into the ureters. So ureters are long tubes, about 25 centimeters to 30 centimeters long tubes, from the kidneys, from the renal pelvis of the kidneys down into the bladder. So they open into the bladder. So here you can see all the three structures. So these tubes that are coming behind the bladder, they're posteriorly behind the bladder, right? So they are the ureters. This bag-shaped structure, like a inverted pear-shaped structure, you can see, that is the urinary bladder. So this tunnel, that passageway, that is the urethra. Now, in females, the urethra is much more shorter, so only about 1 to 2 centimeters, but in men, it's much longer. Now, specifically about the bladder is made up of smooth muscles called detrusor muscles, and these are very, very, very important when we talk about maturation reflex, or the reflex that urges us to urinate. Okay, stretching of these muscles will send a signal to our brain, to our control centers, and they will generate a reflex to relax the sphincters. Now, sphincters, as you know, they are the bands of muscles around the tubes, right? So in case of renal system, particularly when we look at the bladder and urethra, there are two sphincters, like you had two sphincters in the rectum, right? You have the internal anal sphincter and the external anal sphincter. Similarly, for urethra also, you have the internal urethral sphincter, which is smooth muscle band and it's involuntary, and you have the external urethral sphincter. This is a skeletal muscle of the perineum, and it is a voluntary sphincter, right? So we can have some control of over the urge of passing urine because of this external urethral sphincter, which are the muscles in your perineum, right? So that's why pelvic floor exercises are advised when people have incontinence of urine, right? So they cannot control their urge to pass urine. Now this region here, the region between the opening of the two ureters into the urinary bladder and the urethra, right? So these three openings, right? So the region between these three openings, this is called the trigone. This is a smooth bag-like structure, and trigone stretches as urine collects in the urinary bladder. Your urinary bladder has a capacity to store approximately 700 to 800 ml of urine, right? That is its normal capacity. But it can extend way beyond its normal capacity. It can store up to 2 to 3 times more than its normal capacity. It can extend, okay? And that is what is seen particularly when somebody has acute renal failure, or even urinary stones and things like that, and the urine is contained in the bladder. It's not voided easily. The bladder stretches and distends the abdomen and fills up pretty much lower abdomen. Okay, so it can hold up to 3 times its normal capacity. Normal capacity is 700 to 800 milliliters, right? So we talk about the maturation reflex now. So talking about the maturation reflex, it is obviously parasympathetic response, right? So whenever there is a fluid collection in the bladder, particularly urine collection in the bladder, when it reaches its capacity, the stretching of the bladder muscles, the detrusor muscles, will send a signal through the spinal cord into the brain, then that's usually when almost half of the bladder is full, right? This maturation center is located in the sacral segment of the spinal cord, so S2 and S3. Now, if you remember the parasympathetic nervous system is also called lumbosacral nervous system. So the control centers for these reflexes are located in the lumbar and the sacral segments, right? Cerebrosacral. So it's located in the cerebral nerves and the sacral segments, S1, 2, 3, 4. So for the maturation reflex, these are located in the S2 and S3 segments. They will cause the detrusor muscles to contract, and they will allow the contraction of the detrusor muscles. Look at the location of the detrusors, right? So detrusors are located here. So think of what will happen when they contract. When they contract, the bladder is going to contract and the bladder is going to push. As the bladder will push, it will cause these sphincters to open up, right? Because there will be stretching of the sphincters and it will allow the urine to flow out. Okay, so that is the maturation reflex. Stretching of the stretching of the urinary bladder muscles causes the sends signals to the parasympathetic ganglia located in the S2 and S3 segments of the spinal cord, and they will then generate a signal back to the detrusor muscles, causing them to contract. As they contract, the urine squeezes down into the urethra, stretching the internal sphincter, urethral sphincter, and the external urethral sphincter as well. Okay, so male urethra and female urethra, this is something that we will revisit in the reproductive system as well. So female urethra is approximately, you can say, so this is the urinary bladder, the triangle structure here, and this is the urethra. So female urethra is approximately 4 centimeters in length. Compared to that, a male urethra will be about 20 centimeters in length. There is a difference between this because in male urethra is basically also the passageway, not only for the urine, but is also the passageway for semen in men. So which is a produce, semen is basically the secretions from the testes and the male reproductive glands like the prostate and the seminal vesicles, etc. So all that to collect it together along with the sperms, it's all passed through the same structure, which is the urethra. So that's why it is a longer structure. In women, the uterus and its opening are separate from the urethra, right? So uterus, so this bag-like structure here, that's the uterus. This is the vagina, right? And the last part of the uterus here is the cervix, okay? So because there is a separate passageway for the sperms to travel up into the uterus, we don't need that longer urethra here. It's a completely different organ there, okay? Which is not the case in the men, all right? Okay, so that's about the normal structures around the urinary system. Now, quick look at the fluid balances in the body, okay? And I'm skipping a few slides here and there, but it's basically because you can read through them, not quite significantly important. Okay, if you look at the fluid compartments in the body, majority of the fluid, look at how most of the fluid is stored in the intracellular and extracellular parts, okay? So this is a comparison between men and women. Slightly different, like in, for example, women have a slightly less fluid volume compared to men. Well, that's okay. But if you look at the total fluid in the body, most of the fluid in the body, about 2/3 of the fluid, is actually in the cells as intracellular fluid. Whatever is outside, 80% of that is your interstitial fluid, and 20% becomes a part of the blood plasma. So most of the fluid is interstitial fluid, and therefore all the changes that happen in our body are relevant to maintaining that interstitial fluid component. So again, lymphatic drainage becomes very important here to maintain that balance. Okay. So intake and excretion of urine is also, fluid is also very regulated. So this diagram here gives you an approximate comparison of water gain and water loss in the body. You can see that we take in water in many different forms, either through the food or through the drinks that you consume throughout the day. So on an average, a person is expected to consume about 2.5 liters of water. Out of that, if you look at how much is being eliminated, almost equal quantity is being eliminated. Majority of it is eliminated by your kidneys, like 1.5 liter is eliminated through your kidneys. But then your skin, lungs, and GIT also help to eliminate water. Some part of water is eliminated as a part of the stool through the GIT. Some is eliminated through perspiration through the skin, and some is eliminated in breathing when you are breathing in and breathing out. So with the breathing, the fluid and heat is eliminated through the lungs. Okay, but most of the time, majority of the water out of that 2500 ml that has been consumed, 1500 ml, so 1.5 liters of water is actually eliminated by your kidneys. So that's why kidney becomes the most important organ for water retention and water balancing in the body. Okay. So this one here is just kind of helping you to understand what happens when a person is severely dehydrated. Again, a tip here, this will be also very useful for your lab, because a lot of scenarios around your lab might be around dehydration. So you might have to talk about how these different mechanisms can be stimulated in each given scenario. Okay, so whenever there is dehydration, body will try to conserve water with the help of hormones. So which are the hormones that participate? Primarily antidiuretic hormone, right? That is the most important one, antidiuretic hormone. Other than antidiuretic hormone, angiotensinogen, so angiotensin 2 basically, and the atrial natriuretic peptide, they also help to maintain the water balance in the body. So how do they help, right? So when we talk about angiotensin 2 and ANP, the atrial natriuretic peptide, so angiotensin 2 and aldosterone, they work through the renin-angiotensin-aldosterone pathway, and they primarily help to reabsorb water when we are severely dehydrated. So they reabsorb sodium, chloride, and water, and increase elimination of hydrogen ions and elimination of potassium from the body, and thus help to conserve water in the body. Atrial natriuretic peptide helps to promote excretion of sodium and chloride from the body, so it helps to decrease the blood volume. So whenever your heart is under a lot of preload, when the blood volume is very high, or when the body is filled with fluid because of whatever reason, like there is improper lymphatic drainage, or there is renal diseases or kidney failures and things like that, right? So, and not enough water is being eliminated from the body. Water conserves in the body, leading to edema, swelling, right? So in that cases, when there is overload on the heart, like in congestive cardiac failure, right? So in all those conditions, right, the natriuretic peptide from the atria, from the heart, it helps to reset the sodium and chloride excretion by acting on the kidneys. It acts on the PCT and the cells in the loop of Henle and around the kidneys, and helps to eliminate more of the sodium and chloride. It inhibits the reabsorption of these substances in the structures and helps to keep them in the renal tubule so that they can be eliminated. Because we saw that if sodium is reabsorbed, water follows sodium, right? So what we are doing by limiting the reabsorption of sodium and chloride, we are limiting the reabsorption of water and helping to reduce the blood volume. Okay. Antidiuretic hormone, I've already told you about it, so I'm not going into too much detail, but it's basically acting on conserving water in your body by opening up the aquaporin 2 channels in the collecting duct, and thus increasing the permeability of these structures to water. Okay. Now, it is very important to maintain the electrolyte balances in the body as well. Here, the diagram is showing you a comparative levels of electrolytes in the blood and in the urine, sorry, in interstitial fluid and in the intracellular fluid. So basically in the body fluids. Okay. So if you look at the blood plasma, the concentration of sodium in the blood plasma and concentration of sodium in the interstitial fluid, that is in between the cells, is pretty equal, right? Composition of sodium inside the cell is significantly low compared to that what is outside the cell, right? Potassium, the story is opposite. The composition of potassium in the plasma or interstitial fluid, that is in the extracellular fluid, is very, very low compared to that in the cell. Okay, and this has to be maintained, because if you change this, if the amount of sodium and potassium in our body changes, then there will be problems with the electrical signaling. There will be problems with the activation of the cofactors in the enzyme reactions. There will be problems with maintaining the fluid balances in the body. So a lot of things can go wrong here. Calcium, again, calcium is more outside the cells compared to inside the cells. Magnesium, same story, more opposite story, more inside the cell compared to outside the cell. Chloride, again, more outside the cell compared to inside the cell. So this is just giving you an idea of the comparison between the intracellular and extracellular fluids. Okay, so you can see that things like potassium, magnesium, phosphates, proteins, these love to remain inside the cell compared to outside in the extracellular fluid. And it is important to maintain this balance, because these are all important for a lot of electrical activities that are happening in our body, muscle actions that are happening in our body, the co-stimulations of different enzymes happening in our body. All of that is regulated by the electrolytes. So the following few slides will take you through the individual actions of these electrolytes, so you can read them in your free time. Now I want to quickly, because we are running out of time, I just want to quickly touch base with the buffer system. Okay, so when we talk about the acid-base balance, there are three ways in which your body tries to maintain the pH of your blood between 7.2 to 7.3 ish, so maintaining your blood pH into slightly alkaline, right? So it uses the buffers. So when we talk about buffers, buffers are basically combinations of weak acids and weak bases. Whenever hydrogen ion concentration increases in your body uses weak bases like bicarbonates, for example, to buffer it out. Whenever the concentration of bicarbonate increases in our body, right? Like because, say, for example, we have not been able to eliminate enough of carbon dioxide, or there is, excessive metabolic activity producing a lot of bicarbonates and things like that, right? So in that cases, your body will use weak acids, like, for example, carbonic acid or hydrochloric acid and things like that. So they will use acids to balance out the bicarbonates. Phosphoric acid, carbonic acid, these are weak acids that are used by your body. Okay, so when the pH of the blood changes and it becomes more acidic, so when hydrogen concentration in the body increases, that is called acidosis. So I'm just going to take you to the concept of acidosis and alkalosis. Okay, so acidosis is basically when your blood pH is very, very low. So any blood pH which is lower than 7.35 is termed as acidosis. It can be because of respiratory failures, or it can be because of metabolic activity. So there can be a respiratory acidosis, there can be metabolic acidosis. Okay. Respiratory acidosis is when there is a higher concentration of carbon dioxide in body. You're not able to eliminate carbon dioxide, like we talked about when you have reduced lung compliance and you can't breathe out, so you are accumulating carbon dioxide in the body. So that will lead to acidosis. Alkalosis is opposite to acidosis. So alkalosis is basically where the blood pH is more than 7.4, so highly alkaline. and that is when the carbon dioxide levels are very, very low, like hyperventilation, for example. Right? So alkalosis also can be respiratory or metabolic. When we talk of respiratory acidosis, I mentioned that it's all to do with the amount of carbon dioxide in the blood. So increasing the partial pressure of carbon dioxide causes respiratory alkalosis. Metabolic causes for acidosis to happen will be when you are producing too many bicarbonate ions. Okay. So abnormally low bicarbonate in the arterial blood, so you're eliminating a lot of bicarbonate in the urine, and that causes collection of hydrogen ions in the blood, and that is a result for the metabolic acidosis. Metabolic alkalosis is high bicarbonates in the blood, and therefore it results into the collection of bicarbonates and elimination of hydrogen in the blood. Most of the time, metabolic acidosis and metabolic alkalosis are associated with abnormal kidney functions, abnormal liver functions, things like that. So organ failures and things like that. So metabolic diseases like diabetes, for example, they often lead to acidosis or alkalosis. Okay, so in the slides in your session notes, these things are already given to you. And you will, again, these are also some things, concepts that will be useful for your lab again. Okay, so we'll talk about these again in the next session when we talk about the lab, because these are your concepts for the next lab. Okay, so anybody has any other questions to ask me?
No, I don't have any, thank you.
All right, so then that brings us to the end of today's session. So we're going to stop here, and Wednesday we will be talking about the lab. So I will revisit some of these concepts again during that session, particularly the concepts around the acid-base balances and alkalosis, acidosis. This last part of the session, we will revisit that because that is relevant to your lab. Okay. All right. Thank you everybody for joining, and I will see you on Wednesday.